Welcome to The Explainer. Today we are talking about a technology that is literally reshaping our world one gene at a time. This is the kind of stuff that used to be pure science fiction, but you know what? It's very, very real. So just think about that question for a second. What if we could actually program life? Well, that's not a hypothetical anymore. It's the whole promise of genetic engineering. We're going to dive into how scientists are editing the actual blueprint of life and what it means for everything from brand new medicines to the food that ends up on our plates.
Now, this whole idea has a pretty scientific name, recombinant DNA technology. But at its heart, the concept is surprisingly simple. You take a piece of DNA from one organism and you stick it into another. But that one simple action, it has given us this incredible, almost mind-boggling power to redesign life itself.
Okay, but before you can even think about rewriting the book of life, you first have to learn how to read it, right? So the first giant leap was cracking the universal code that all life is built on. You can think of DNA as a kind of universal operating system. It's wild, but it's written in the same fundamental language whether it's programming a tiny microbe or a person. And this shared language, this common software, is the key that makes genetic engineering even possible. And what's really incredible is just how simple this language is. It only uses 4 chemical letters, and these letters are grouped into 3-letter words called codons, which you can see right here. Each one is a specific instruction. It either names a building block for a protein, or it just tells the whole process when to start, or, you guessed it, when to stop. Simple as that. So, understanding the code was a huge deal, but editing it, that required a whole new set of tools. Let's open up the molecular toolkit that really brought genetic engineering to life. The first massive breakthrough was finding these natural proteins that could act like unbelievably precise scissors. We're talking about scissors that don't just cut anywhere, but at very, very specific letter sequences in the DNA code. And these molecular scissors have a name. They're called restriction enzymes. They give scientists the power to target and snip out a specific gene from a super long strand of DNA, isolating the exact piece of code they want to work with. Okay, so you've used your scissors and cut out the gene you want. Awesome. But here's the problem. One single copy is pretty much useless. To do anything meaningful with it, you need millions, maybe even billions of copies. So what scientists really needed was a genetic photocopier. And the solution they came up with is a process called the polymerase chain reaction, or PCR. It's a really elegant 3-step cycle. So first, as you see in step 1, you use heat to denature or unzip the 2 DNA strands. Next, in step 2, these little things called primers lock onto the target sequence you want to copy. And finally, an enzyme gets to work building out from those primers to create a perfect copy. You just repeat that cycle over and over, and with every single round, you double the number of copies. It's an exponential explosion of DNA. So you've cut out your gene, you've made billions of copies, now what? How do you actually get it inside a new organism? For this, scientists turned to nature's own delivery systems. These molecular delivery trucks are called cloning vectors, and pretty ingeniously, scientists realized the perfect vehicle already existed in nature: plasmids. These are these tiny circular pieces of DNA found in bacteria, and they are absolutely perfect for carrying a new gene into a host cell. All right, so we've read the code and we've assembled our toolkit. Now for the really cool part. Let's see how all these pieces come together to actually build a brand new organism. We'll use a transgenic plant as our example. Let's just walk through how this works, step by step. First, scientists insert the gene they want into one of those plasmid vectors we just talked about. Second, they transfer that plasmid into a special bacterium called Agrobacterium, which is basically nature's own genetic engineer for plants. It's amazing. Third, that bacterium infects the plant cells, and it naturally transfers the new gene right into the plant's own DNA. And finally, from just one of those transformed cells, you can grow a whole new plant, a plant that now carries that brand new trait in every single one of its cells. And look, this isn't just theory or something that happens in a lab. Genetic engineering has moved out of the lab and straight into our daily lives. It's creating products that have a real, tangible impact on all of us. The applications are just incredibly diverse. We're talking about fundamental changes to agriculture, totally new ways of manufacturing life-saving drugs, and even developing brand new medical therapies. It's a game changer. I mean, just look at the effect on agriculture. It's been massive. Take insect-resistant crops, for example.
The data here shows that between 1996 and 2013, this technology boosted average maize yields by over 10% and cotton yields by a pretty staggering 16%. That's a huge deal.
And the applications go way beyond just yield. We have tomatoes engineered to ripen more slowly, which extends their shelf life. In medicine, we've created recombinant vaccines. And get this, we've even created farm animals, like these transgenic pigs that can produce a human blood clotting factor in their milk to treat hemophilia. Just incredible.
The ability to directly edit the code of life is easily one of the most powerful technologies humans have ever developed. And, as you know, with great power comes great responsibility, and some really important questions we have to ask ourselves.
You know, as our tools get more and more precise, from those early restriction enzymes to newer technologies like CRISPR, the scope of what we can do is just expanding at an incredible rate. And that forces us to stop and ask, what should we do?
This whole thing forces us to grapple with some huge questions. How can we be sure these organisms are safe for us and for the environment? What happens if an engineered gene escapes into the wild and creates a so-called superweed? And how do we even regulate this stuff? Do we focus on the final product or the process used to make it? And maybe, underlying all of that is the biggest challenge of all. How do we as a society even begin to talk about these complex risks and benefits? When you get right down to it, genetic engineering is a tool. And the story of how that tool will be used, well, it's still being written. Its next chapter really depends not just on scientists, but on the choices all of us make. So what kind of future are we going to build with it?